I'm Christine Curcio, and I thank you all for listening to this tutorial today. This is the fourth of a set of 4 on deposit-driven AMD. Here are my supports and disclosures. The overall key takeaways from the 4 tutorials are summarized here. That AMD is a solvable 4-dimensional puzzle visible in vivo via OCT-anchored multimodal imaging. The photoreceptors and the RPE together form a sensory chip of the brain, with geometry of the foveal singularity being extremely important. We discussed the value of a 3-layer versus a 5-layer Brooks membrane. The essential AMD pathology is extracellular deposits on either side of the RPE. Drusen follow the cone vision system, and subretinal drusenoid deposit begins where rods are numerous. We think the lipid composition of the deposits hints at a major system to feed the membranes of outer segments. To review, the OCTB scan on the left shows the layers affected by AMD. The schematic shows the progression from normal aging on the left to advanced AMD on the right. The reflective bands arise due to the precise horizontal alignment of millions of vertically compartmentalized cells. There are multiple biologically distinct layers within 100 micrometers vertical height between the ELM, the dashed red line on the top, and the choriocapillaris endothelium below. These include the inner segments of the photoreceptors, the outer segments, the interdigitation zone, where the outer segments and apical processes of the RPE meet, the RPE cell body layer, and Brooks membrane choriocapillaris together. These bands may be visible, perturbed, or absent altogether in an AMD eye. Layers are added representing the extracellular deposits, SDD between the photoreceptors and the RPE, basal laminar deposit between the RPE and its native basal lamina, and drusen and basal linear deposit, 2 forms of the same material between the RPE basal lamina and the inner collagenous layer of Brooks membrane. So basal laminar deposit, or BLamD, is the subject of this tutorial, and this represented the first histologic staging system for AMD, with its abundance associated with increased risk for progression in the seminal work by the Sarks-Killingsworth group. It's now visible in OCT. It represents an aberrant form of RPE basal lamina, present in all AMD eyes and not unique to AMD, as it appears in several monogenic inherited disorders. It is a prerequisite for high-risk drusen, and it traps soft drusen material in streaks and in basal mounds. And we can see a path to valid mouse models relevant to drusen in animals that have BLamD. The terminology for BLamD and BLamD has been confused by investigators viewing them at different levels of resolution. We settled on the terminology used by the Sarks based on transmission electron microscopy and agreed upon by Dick Green in the US and the Rotterdam group in Europe. I apologize for using the term basal deposits in 2005. This term is unwarranted if high-resolution histology is used. W.R. Green, the pathologist, emphasized the importance of understanding that these 2 deposits, basal laminar and basal linear, for prevention of AMD. Shirley Sarks described 6 stages of AMD based on BLamD abundance, starting with aging on the left and ending with fibrovascular scars due to neovascularization on the right. It was especially prominent in intermediate AMD and stage 4, and persistent in atrophy, stage 5. Now, her judgments were based on a large sample of clinically characterized eyes from residents for whom she was the ophthalmologist in a retirement home. She used state-of-the-art paraffin section histology and principally the Picro-Mallory trichrome stain, which stains 3 tissue elements. As you can see, the photomicroscopy and journal reproduction quality has continued to improve since this seminal paper in the mid-70s. We can think of Brooks membrane as a flat vessel wall between the choriocapillaris on the bottom of this picture and the RPE on the top. Our studies have led us to agree with Sarks and with Gass that AMD pathology is best explained if we think of Brooks membrane as its middle 3 layers rather than the traditional 5 layers. The RPE, being an epithelium, has a basal lamina with different collagens from those found in the other layers. And in AMD, the basal lamina thickens to become BLamD. It adheres to the RPE and opens a cleavage plane called the sub-RPE basal laminar space, where we get drusen, type 1 neovascularization of choroidal origin, and fluid. Different monogenic disorders affect different sublayers of this complex, and notably the genes TIMP3, C1q, TNF5, and FIB3 give rise to phenotypes with a thick basal laminar deposit. This slide shows well that BLamD is internal to and distinct from the basal lamina. This is the native basal lamina in a very well preserved eye. Banded type 6 collagen is apparent, as is a homogeneous material of 2 different electron densities. The Rotterdam group provided important and comprehensive light microscopic histology of 182 consecutive donor eyes. In the central retinal area, BLamD increases with age in the bottom graph. And they showed these stages of BLamD, which is patchy, thin continuous, and thick continuous, where thick and thin are referenced to the height of healthy RPE cell bodies. The general composition of the RPE basal lamina can be inferred in part from other epithelia, as direct comprehensive assays are still lacking. These include collagen 4, laminin, nitogen, heparin, sulfate, proteoglycan, laminins, collagens of different types, fibulins, and dystroglycan. Due to low solubility, it was difficult to make specific antibodies to extracellular matrix proteins. However, antibodies to specific collagen type 4 chains that were created by recombinant technology did reveal that the RPE basal lamina is similar to those in the glomerular basement membrane of the kidney, another basal lamina involved in filtration. Whereas other type 4 chains also localize to the choriocapillaris basal lamina and to major vessels. Bovine RPE basal lamina has laminins A5, B2, and nitogen 1. Immunohistochemistry was used to specify extracellular matrix proteins in human specimens with BLamD. These, plus vitronectin, ECM regulators like metalloproteinases, and complement proteins were also found. An EFEMP1 knock-in mouse with thick BLamD was used for proteomics of a combined BLamD and Brooks membrane preparation and identified these and more. A classic stain that was used by the Sarks has been a workhorse for us, and this is periodic acid shift hematoxylin, or PASH. It stains carbohydrates in Brooks membrane, BLamD, and drusen. Be aware that a routine hematoxylin and eosin stain does not stain BLamD. A fluorescent marker for these carbohydrates would be an important new tool if such existed. To investigate AMD deposits, including BLamD, we created a website of AMD pathology called Project Macula, with 132 short postmortem eyes collected over 14 years. These were processed for high-resolution histology, which is essentially an electron microscopy prep viewed at light microscopy levels. Sections through the fovea and the superior perifovea were systematically reviewed, photodocumented, and uploaded. The thicknesses of chorioretinal layers and the contents of subretinal and sub-RPE basal laminar spaces were measured and annotated at predefined locations shown below. The histology and sampling scheme and the photomicroscopy were all designed under the hypotheses that the photoreceptors drove the metabolic demands on the vascular support system, and that the signal source for OCT was subcellular. Here are different forms of BLamD from the website. Here's the normal basal lamina, which is not visible at this magnification. Early or patchy BLamD. Here are the two thicknesses. Early is at the bottom, late is on top. What came out first from the RPE is at the bottom, like pouring a beer. BLamD can outlast the RPE when it dies. Here it persists in geographic atrophy, and it was important clinically to learn that the hyporeflective space under that bright line was not fluid and did not require treatment. BLamD also persists past RPE death in neovascular AMD. Here is an analysis of layer thicknesses from the website. BLamD separates AMD well from normal. It's ubiquitous in AMD eyes. RPE thickness is stable across all stages if atrophic RPE is not considered. Defined by its three layers, Bruch's membrane is stable in thickness and actually thins in neovascular AMD. And finally, RPE and basal laminar deposit together thickens with progression, moving the RPE even further away from the chorea capillaris. We were fortunate to study the eye of a patient who had been seen clinically 14 months before death. The OCT scans show a split fourth band or shallow irregular RPE elevation called a SIRE. This is also called a double layer sign. And the histology showed that the hyporeflective band could correspond to either BLamD alone in panels E and F or in type 1 neovascularization with BLamD in panels G and H. One might be treated on the right, and the one on the left would not be treated. New OCT technology may show even more details of BLamD. This device uses visible light technology to achieve 3 micrometers axial resolution. These scans through the fovea are collected on this radial pattern, and below the highly reflective RPE band is a hyporeflective band resembling BLamD over confluent soft drusen. Basal mounds is a Sark's term to denote soft drusen material trapped within basal laminar deposit. These form because lipoproteins released by the RPE are blocked in their transit to the chorea capillaris by RPE basal lamina. The basal mounds can be focal or diffuse, and if thick, they can elevate the RPE and be mistaken for drusen. They can also persist after the death of the RPE and calcify. So this material undergoes the same mineralization pathway as soft drusen material in the sub-RPE basal laminar space. Since my AMD world is drusen-centered, we used Philippine histochemistry to label unesterified and esterified cholesterol in BLamD. At the left, arrowheads delimit thick BLamD with both UC and EC preferentially at the base, near the intercollagenous layer of Bruch's. In the Project Macula eyes, basal mounds preferentially localized under the fovea, as do other soft drusen materials in both AMD and aged eyes. We used lipid-preserving ultrastructural techniques to demonstrate solid electron-dense material distinct from the membranes of RPE basal foldings crossing BLamD. BLamD may be used to not only stage human AMD, but also to develop drusen-relevant pathways in mice. Lipoproteins from the RPE, and I refer you back to the overview and to the tutorial on drusen. These lipoproteins must cross BLamD to enter the sub-RPE basal laminar space. This case of late-onset retinal degeneration, or LORD, caused by mutations of the C1q-TNF5 gene, gave us the best opportunity to study lipids crossing BLamD. The classic stains showed carbohydrates and phosphates that were part of calcifications. Oil red O and philippin for esterified cholesterol are confined to the deposit and are most intense along the basal border, and the deposit contains apolipoprotein B, the principal protein of a large lipoprotein made by the RPE. If you read this paper, look at the figures. Don't read the discussion, because this predated a formal model of RPE as a lipoprotein secretor. On the right, compare standard osmium post-fixation on the left for extracellular lipid to the osmium tannic acid paraphenylene method, which was developed for atherosclerosis research. The OTAP method shows strings of particles crossing the basal laminar deposit. BLamD also appears in aged mice. Here is a transverse section of basal RPE showing the basal infoldings in animals up to midlife. These are especially apparent in the transverse plane below. We estimated in humans that the basal infoldings expand the absorptive capacity of RPE tenfold over what a flat membrane in the same area would provide. In older animals, amorphous material appears between the enlarged individual infoldings. This loss of membrane brings with it loss of transporters, receptors, and channels expected to impact RPE metabolism and ability to stain photoreceptors. With this background in human pathology, it's possible to see similar phenomena in aged mice of different strains created by different manipulations, including diet. So BLamD in mice appears to be a marker of RPE stress. Here is collagen type 18 deficient, 2 different EMP1 knock-ins, CFH risk allele heterozygote, a high glycemic diet. Here's a kinesin knockout with 2 different EM preparation techniques, like what I showed before. This showed a positive evidence that the wholly features are indeed lipid. So this brings up an important point. To check your mouse model with a transmission electron microscopy like they did, or with lipid histochemistry like Goldus Malik's lab did. These investigators found electron-dense material in the same compartment as oil red O signal. And this is especially important because in some situations the lipid is not neutral lipid at all, but rather membranes of the surrounding cells. This model of LORD, produced by an adenovirus vector, has a thick BLamD visible in OCT and histology. And here it is by electron microscopy. Immunofluorescence for the GLUT1 transporter, a signature basal infolding protein, is expressed throughout the deposit. To quote Astrid Dinculescu, the RPE powers the height of the deposit by essentially growing with it. And this contrasts with aging human AMD and LORD, where the basal laminar deposit replaces the RPE infoldings. To summarize this tutorial of BLamD, this is the first histologic staging system for AMD, which showed that abundance was associated with risk of progression. It's now visible in OCT. It's an aberrant form of RPE basal lamina present in all AMD eyes, and it's not unique to AMD, as it occurs in monogenic inherited disorders, which has enabled us to get new promoters and new tools. It's a prerequisite for high-risk drusen. It traps soft drusen material within it as basal mounds, and a path to mouse models relevant to drusen is available through different animals that have BLamD. Here again are the key takeaway points from the 4 tutorials, and I thank you for your attention today.